Hi, I'm Amy Ramsey. I'm a professor at the University of Toronto, and I'm a researcher who studies GRIN disorder. It's a rare neurodevelopmental disorder caused by variants in the genes that make up the NMDA receptor. Before I tell you what our lab does, please let me introduce you to our lab. Um, I work with an amazing team of scientists who are undergraduate students, graduate students, postdoctoral fellows, and research associates. And they are the ones who are working hard to try and help children with GRIN disorder. We work with mouse models, and we can use these mice to help children with GRIN disorder by studying the patient variants, making the mutations in mice that copy what the children have. We can test new and existing drugs for their ability to improve the symptoms. And we can also test emerging gene therapies for their ability to improve the life and health of these children. Some of the mouse models that we work with carry different point mutations in the GRIN1 gene. And we have mice that have overactive NMDA receptors, and we have mice that have underactive NMDA receptors. And we study these mice in a lot of different ways. We look at their brains and we look at their behaviors. We study how they interact with other mice. We study their preference for open spaces versus closed spaces. And we study their ability to learn and remember different maze configurations. So all of these can be used to test therapies and determine whether they're actually going to be beneficial for children. Excitingly, we're starting to work with a small biotech company that makes gene therapies for humans, and they have agreed to help our lab develop gene therapies for children with GRIN disorder. And to do this, we're going to take medicines that they make and put them into our mouse models. Then we'll be able to study the different cells of these mice, and we'll be able to study the different organ systems. And of course, we're going to be able to measure their behaviors to see whether it actually improves their ability to learn and remember, communicate, interact with other mice. And hopefully this will be the first step towards making gene therapies for people. I want to say thank you to all of the families and all of the children especially who have taught us so much and give us the motivation to keep working hard. I also want to thank the research foundations and the government support from Canada that have enabled our research. Thank you so much for your donations and for your funding.